Welcome to Science Frontiers, a space for discussions in the frontier areas of science and technology. I am your host, Binay Panda. Synthetic biology, while bringing much joy to the scientific community, also brings fear. The fear for creating something new that has long been thought to be an exclusive domain controlled by the creator, the God as some believe. This brings the students of theology and biology at loggerheads. In fact, a few years back, 2017 to be exact, in an op-ed article for The Hindu, I had mentioned this and how some believe that using synthetic biology techniques, scientists may claim to play God. Well, what is the reality? Can human beings play God? And are we at a time when we can claim that we have created living organisms in the lab? Not getting into the scientific details, it is important that we as a society build mechanisms to regulate experiments that may be considered dangerous or unethical. The most important point is not to get to that point and come up with mechanisms that the public at large can be assured that the experiments that the scientists do and conduct in the lab pose no danger to humankind, while at the same time encouraging scientists to innovate in the lab. Today's discussion is exactly about that, the policy parameters that we have and we need to have for synthetic biology. We shall discuss about this with today's guest, Pawan Dhar. Pawan is a professor in the School of Biotechnology, Jawaharlal Nehru University, New Delhi. Pawan, welcome to the forum and thank you for joining us. As you know, there are multiple international organizations and mechanisms in place to regulate experiments in synthetic biology. In fact, many of them are broad to regulate all experiments that involve recombinant DNA technology. In fact, India, as the signatory of all the major international conventions that regulate biological experiments related to DNA, are abiding by that. For example, the Convention on Biological Diversity, or CBD of 1992, the Cartagena Protocol on Biosafety to the Convention on Biological Diversity 2000, or what we call as Cartagena Protocol in short. And finally, the Nagoya Kuala Lumpur Supplementary Protocol on Liability and Redress to the Cartagena Protocol on Biosafety, or CPB. In India, too, we have several acts, regulations, rules, guidelines, and policy documents that govern biological experiments involving recombinant DNA. Some of those are the Environmental Protection Act of 1986, rules for the manufacturing, use, import, export, and storage of hazardous microorganisms or genetically engineered organisms or cells of 1989, Drugs and the Cosmetic Rules of 1988, Schedule Y of Drugs and Cosmetic Act, Protection of Plant Varieties and Farmers' Rights Act 2001, Biological Diversity Act 2002, Food Safety and Standards Act 2006, and the Plant Quarantine Order 2003. In fact, in addition to these rules and regulation, we have several policies, guidelines, protocols, and notification. I'm not going to go into that detail. If any of you who are interested to know more, kindly visit the Indian Biosafety Knowledge Portal of the Department of Biotechnology, Government of India. So as you can guess, there is a lot of confusion on the policy. If you add this to what already exists, particularly adding the Institutional Biosafety Committee or the IBSC, the Review Committee on Genetic Manipulation or RGCM, and perhaps the most important among all, the Genetic Engineering Appraisal Committee or GESE, which is one of the most important committee of the Ministry of Environment, Forest and Climate Change. GESE is very important because as per the rule of 1989, it is responsible for appraisal of proposals relating to the release of genetically engineered organisms and products to the environment. Now, Pawan, with all that in the background, we have so much in terms of policy, guidelines, notifications, rules, and regulation. Why do you need something specific or even more for synthetic biology?
Thank you, Binay. I think this is a very important topic because the issue of synthetic biology governance has been in what I call in suspended animation for more than 18 years, ever since the field was formally launched. Well, the existing system of regulation, manages the tinkering of genomes or translocation of genomes. It could be point mutations, it could be deletions, it could be lifting the sequences from one organism and expressing in another organism. However, synthetic biology offers you at least 3 new challenges to my best understanding, which are not covered in the existing regulatory system. First is the long DNA synthesis, your ability to synthesize a gene or a full genome. And second is making a non-natural DNA, which was not designed by nature. For example, in 2008, Japanese scientists from University of Toyama, they came up with a non-natural basis, creating an artificial DNA. Now, this DNA does not transcribe or replicate, but it looks like the original DNA that we are familiar with. And the next step is definitely to make it function. Likewise, the third aspect is the genetic code expansion. In 2017, scientists from Scripps made a 6-base DNA, adding X and Y, and this is a functional DNA. And it has led to what people popularly called as the hacking of genetic code. So from the given set of canonical amino acids, we have non-canonical amino acid synthesis, raising the numbers to more than 200. So this calls for clear-cut policy guidelines.
So excellent, Pawan. So you mentioned about 3 things. One, we need specific policy intervention because we are talking about longer DNA synthesis and DNA synthesis that can actually expand to whole genomes of the organisms. Number 2, you talked about the policy intervention requirement for non-standard nucleic acids or amino acids in proteins. And number 3, for unique functions. And that's very good. Now, give us an example that how a specific policy intervention can make things very clear for the field.
Thank you, Binay. Well, you see, to design a policy, first you need clarity on the definition. If you tomorrow say that what we do is illegal, then I need to know what is legal. So to define what is legal, synthetic biology field needs a crisp definition and an exclusion criteria. Right now, the field itself is blurred by the lack of a standard definition. And though scientifically people understand this as the construction approach in biology, but if you look at the definitions that have been proposed by the regulatory agencies, they are immensely overlapping with the existing recombinant DNA technology. So the first and foremost is clarity of the definition. Second, we do not have a clarity on building the standards, composing the devices and circuits. There's no clear policy on that. Suppose I take a part from a virus, a deadly virus. The part itself is not deadly, but it's coming from a deadly virus. Am I allowed to compose something from this? Maybe I'm coming up with a non-pathogenic variant, which is not the whole virus, but a subset of the virus. Is it possible to do that? What is the policy on that? We just went through the expansion of genetic alphabets. We do not have a clear-cut policy on that. This is very specific examples I'm citing. Currently in India, at least in our country, we don't have a study on the horizon mapping, foresight, and forecasting. There's hardly any data. Well, in addition to that, there is also efforts going on, a lot of discussions going on, whether we should bring back the extinct species back to life. So in a way, de-extinction. So what is the policy on de-extinction? Should we bring back the woolly mammoth dinosaurs, or should we bring back the recently extinct species, or maybe the endangered species? Well, there are scientific merits for all of these case scenarios, but what is the policy? That's lacking. even though synthetic biology got a bad press, but the number of practitioners are increasing, as you know. Globally, people are adopting this, in a big way, because of which the biosecurity aspect is also coming into light. See, today you have technology by which you can synthesize a deadly virus. Not only that, what is more alarming is that people have even published the protocols. The deadly polio virus that has been, an infective deadly polio virus that has been created in the lab, people have published protocols in one of the very good scientific journals. Should this be allowed? How much of sharing is too much? And how much should remain within the labs? Because we are talking of responsible research or responsible innovation. And there are a couple of points more here. One is that when we talk of synthetic biology, people get excited, and you have these DIY initiatives when people want to make their own devices or on their parts. So should we also not regulate the non-specialist biologists from tinkering the genomes and the organisms? And people have used terms like unintended consequences or unpredicted events, especially in relation to synthetic biology, which I feel very strongly that this is unfair, because these unintended consequences or unpredicted events can happen in any field. You tinker the existing genomes, and you can also have unexpected consequences. So synthetic biology need not be looked at as an evil field that has something monstrous consequences. So in my view, an effective and comprehensive synthetic biology governance may be considered at least three levels. One is the in vitro, wherein one is to be I will not say relaxed, but conforming to the existing recombinant DNA guidelines. But the next level would be in vivo synthetic biology. And the most stringent aspect needs to be related to the environmental synthetic biology. Maybe it's time to have biodiversity commons, but it's important to ensure that biodiversity commons does not end up as facilitator for free riding and piracy. So these are some of my views about the policy plugins that are needed, and we badly need them, and we don't have much time.
Thank you, Pawan, for the specifics. And I think, if we just revisit for a second some of the points that you mentioned, you talked about the need of specific policy interventions because there are protocols, methodologies, and exact reagents required to produce an artificial virus, because the sequence is published, so one can produce that. Number two, you talked about there are efforts undergoing elsewhere in the world on resurrecting the dead animals. In fact, if some of our viewers do not know, I would recommend the book is right there. And I can show you the book by George Church and Regis about regenesis, where George talks about very passionately, and I know speaking with him in person, that the efforts which are going on in getting the woolly mammoth back into real life. Now, we can debate whether it's the right thing to do or the wrong thing to do, but scientifically it's a very exciting thing to do. And I think as long as the point you are making, as long as we have the right regulation in place, we should not really get bogged down of thinking synthetic biology as the evil force. In fact, it can be a good force to deal with as long as the experiments are regulated. Now, let's just go back to another aspect, particularly in the context to India. Recently, Department of Biotechnology, Government of India has actually published on their website a very lengthy document on synthetic biology called Foresight paper. And you are one of the principal investigators, rather the principal investigators for the project, which actually got this document out for everyone. Now, can you tell us a little bit about the specific recommendations that you're talking about in the Foresight paper? And what do you actually see come to the light in a realistic term in maybe in the next 1 decade?
Thank you very much, Vinay. This particular study that you're referring to, the DBT funded study on the synthetic biology policy aspect, It was initiated by Dr. Balakrishna Pisupati, who is the consultant for United Nations, and he is big-time expert in the science policy domain, in particular the synthetic biology aspect. So he introduced me to this idea of doing a foresight paper in synthetic biology. So we worked together on this, and we had several meetings across the nation, including the academia, the students, the scientists, and industry people, and also the administrative people, the science administration. And so we had several rounds of meetings. We wanted to get an idea of what does the community think of synthetic biology in terms of the clarity of this definition. What are the various tracks? So we made different tracks, let's say agriculture, the health sector, the fundamental science, and so on. And each track, there was a group leader who was assigned the job of managing the track and getting the feedback from the group. So it was a comprehensive work done for close to 1 year. And then we submitted the policy framework document. I would say something like 70 to 80% of the policy aspects are already there. We cannot call it a policy document simply because it's a framework. On the top of this, government needs to now invite more people, more stakeholders to examine our recommendations, and also to see what are the specific ways by which we can add data which is India specific. So the recommendations that we had given were many. Of course, one was the horizon scanning, the horizon mapping in India, because the data that we have currently is very little. People may call themselves a synthetic biologist, but are they really working in this area or not? That is not very clear. So the definition part is very, very important. And the second is that the recommendation was that, building the standards. And if you tomorrow have to fund a project, and the project is about taking the, let's say, the parts from the pathogenic virus, and then building a non-pathogenic construct. What is the policy of the government in that? Because now we are talking of construction in biology, the construction approach, in other words, engineering approach in biology. So when you're talking of engineering approach, you have to have clarity on the standards, and the standards of 3 levels: part, devices, and circuits, and then rules of composition. So these recommendations were made, the gaps were identified, and the list of the points that I mentioned a while ago, whether we should bring back the extinct species to life. Will the government of India allow such a project to be funded? What is the policy of government of India? So these points were raised in the document, and this was close to 85 pages. It was submitted to the government of India, the Department of Biotechnology, building a framework. But now this framework is only the foundation. And now we need to have more discussions from different stakeholders, which we may have missed because a lot of people are now active in the field. So now they need to examine the document, and government needs to take a very active approach, and then a certain policy has to be made. And what is really lacking in the Department of Biotechnology at the moment is that we don't have a task force in synthetic biology. So that's another thing that we had mentioned, but I guess they're aware of it and maybe they're working on it.
Excellent. So I picked up one very important thread from what you just told, which is we need very clear-cut guidelines to do experiments and clear-cut policy to do PDC, that is parts, devices, and circuits. So if we know as an experimental biologist that how to deal with parts, how to deal with devices, and how to deal with circuits, and most importantly, if we write a research grant proposal with any of these or combination of one of these, how is it going to be looked at? not in terms of getting the money, but in terms of the regulation and policy. So I think it will be very interesting to take the document that you have produced to the next level and have a much wider and open discussion. Because one of the things also in our country we miss is public participation. Because anytime we talk about biotech or biology related policies, the public needs to be involved. And in the West, there are mechanisms of town hall debates, for example. And I think it's time that we actually educate our people, our public at large, and tell them what we are doing and involve the stakeholders so that they have a say how the policy is shaped. Now, going back to our last question, it's very interesting. Recently, you must have seen the news that the Ministry of Environment, Forest and Climate Change, after receiving the recommendations from the Department of Biotechnology, Ministry of Science and Technology, Government of India, has exempted what they call as SDN1 and SDN2, and SDN stands for site-directed nuclease. So the government has recently exempted site-directed nuclease 1 and site-directed nuclease 2 genomes from Rule 7 to 11 of the Environment Protection Act. Now, for those who are not used to this terminology, in simple terms, basically what government has said that if you have used site-directed nuclease 1 and 2 and there are no exogenous DNA or external DNA in the organisms is going to be exempted from the biosafety assessment. And in other words, the long process of approval that the genetically modified crops of the GMOs go through the Genetic Engineering Appraisal Committee that I mentioned before, GEAC, is now exempted if you have done gene editing using both site-directed nuclease 1 and site-directed nuclease 2. Now, can you, Pawan, tell our viewers, how is this related, if at all, to what we just talked about, synthetic biology policy?
Thank you very much, Vinay. It's a very encouraging moment for us because when you say engineering, engineering means construction and editing. If you have to repair something, this is also engineering. If you have to construct something, it's engineering. So part of the government efforts to allow the genome edited plants without going through the cumbersome GMO regulation, it's a welcome news for us. As you just mentioned, the SDN1 and SDN2, they are exempt from this, the whole process, because one is about introducing the changes, which is already taken care in the standard format of regulation. And the other one is that if you delete a piece of DNA, you add the complementary DNA to this, so it looks like natural DNA. So in a way, I would say it's not a major step, but then it's conceptually, it's a good step forward, kind of a progressive step. Right. So now the question here is, what can we learn from here? Well, I think, I see this as a non-invasive approach towards policy making. So let us say, if government thinks along the same lines and says that, okay, let's allow the long DNA synthesis of natural DNA from non-pathogenic organisms as a clear statement, because still that has not been put on paper. It may be conceptually considered so, but it has not been put on paper. So in a way, the SDN1 and SDN2, they also have a plug-in into synthetic biology because the editing is considered as part of the engineering. It is not separate from engineering. So one can say that we have two plugins imported into the regulatory aspect of biological engineering. So now, if we think along these lines, then as I just mentioned, the long DNA synthesis of natural DNA, that could come as the easiest regulatory approval. The second would be a combination of long DNA synthesis and SDN1 and SDN2. If suppose somebody wants to perform these two experiments together, they should also be allowed because there are already regulatory approvals for SDN1 and 2. But moving on, government needs to really have very clear-cut guidelines about synthesis of the pathogenic microbial genes and genomes. They should have the highest stringency in regulatory approvals. Well, that's the natural step after this. The question is that if I take, let's say, BSL-3 organism, and I remove that portion and make it non-pathogenic, or I'm adding a pathogenic block, how is it looked at from the regulatory perspective? Because this is also the editing part. Or if I synthesize, go for a long DNA synthesis of a organism, and I do not know that a combination of genes from many different sources, some of them are from pathogenic sources, some of them are from non-pathogenic sources. What would I like to call this project? Is it BSL-2, BSL-3, BSL-4? What is it? So those are the plugins that are needed, and that's what we have been able to convey to the government to discuss this aspect. But at least from the approvals perspective, from the plant genome editing perspective, I would say that these two aspects, the SDN1 and SDN2, they can be straight away imported into the synthetic biology regulatory process, and from there we can take few steps forward.
Indeed, I think this is definitely a very encouraging news for the community of exempting SDN1 and SDN2 derived gene-edited organisms. And I'm afraid, Pawan, that's all the time that we have today. And thank you for giving us enough food for thought on the additional plugins that not just the government, but we as the stakeholder, the scientists and the public at large need to think and decide. With that, not only we would like to thank you for the episode, but also thank you for being with us for the entire series on synthetic biology. We wish you good luck for your research, and goodbye.
Thank you very much, Vinay. It was wonderful talking to you, and we look forward to more interaction. Thank you very much.